Good day everyone. This is Dr. Baromeo. On behalf of the Department of Laboratory Medicine, let me welcome you to the Department of Laboratory Medicine. Your first lecture begins with first few chapters of the textbook Microbiology by Joetz. Please try to acquire a copy of the book, and I am encouraging everyone to read the chapters before attending the class. Why study microbiology? 80% of admissions are infectious cases. Most of these are preventable. Scope of the subject: basic science of microbes, some clinical syndromes. Sequence of teaching: basic lecture, laboratory, case studies or journal appraisal. So the objectives for Lecture 1 are as follows: give overview and scope of the subject microbiology, describe the structure of the bacteria, discuss Gram staining, give methods in the classification of bacteria and describe the growth curve of bacteria in vitro. The intended learning outcomes are: the learner by the end of the lecture should be able to differentiate prokaryotic from eukaryotic cell, describe the structure of the bacteria, give functions of its parts and their role in the development of disease, understand how bacteria are classified and grown in the laboratory. The organisms covered by the subject are viruses, prions, prokaryotes or bacteria, and eukaryotes like algae, focusing on dinoflagellates or red tide blooms which produce neurotoxins, fungi, a lot of them, protozoa, which is given in parasitology, and slime molds. This is the image of the virus now causing the pandemic, COVID-19. At the surface of the spherical virus are spike proteins which facilitate viral entry into the pneumocyte type 2, causing inflammation and fibrosis of the lungs. Beneath the spike proteins is a layer of capsule acquired by the virus as it goes out of the host cell by budding off. This membrane is soluble to alcohol and detergent, the reason why we have to wash hands all the time so that the virus is destroyed, or we can use sanitizer to destroy the capsule, therefore killing the genetic material deep inside the virus. COVID-19 has RNA genome, an image of a typical bacillus which we will discuss in detail later in the lecture. There are two types of prion molecules, a normal one and an abnormal molecule at the right side of the image. Prions are proteinaceous materials devoid of genetic material, and they cause a lot of syndromes we don't fully understand, like Kuru of New Guinea and mad cow disease. The normal prion molecule is found at the tip of neurons, while the abnormal prion, once ingested, recruits normal prion molecules to be converted to abnormal prions, folding and refolding the normal prion so that it produces a blockage in the transmission of impulses in the nervous system. This is a typical alga. An image of a dinoflagellate. This microbe eaten by the shellfish, and in the shellfish this microbe produces a neurotoxin when ingested by man. This is manifested as paralysis, slime molds with its life cycle. Sometimes this is also called a schizophrenic microbe because in its life cycle, it assumes the morphology of a fungi, then it becomes a flagellated organism like a protozoon or an amoeba, ultimately becoming a plasmodium which later matures into a fungal-like structure again. Slime molds are sources of new antibiotics, but most of them are edible. Here are hyphal elements of fungal colonies grown in a culture medium. Most of these are common in tropical areas, causing skin problems and opportunistic infection in the immunocompromised patients like HIV-infected individuals. This is a bar chart showing incidence of fungal infections in Asia. A favorite topic or item in any examination that you will take is the differentiation between a prokaryotic cell and eukaryotic cell. The major criterion is the presence of the nuclear membrane in the eukaryote and its absence in the prokaryote. Bacteria have one chromosome while eukaryotes have more than one. Except for ribosomes, prokaryotes have no membrane-bound organelles. Prokaryotes divide by binary fission while eukaryotes by mitosis and meiosis. Both have extra chromosomal elements like the plasmids which carry resistance factors, especially to antibiotics, so the typical bacterium has this morphology. From the outside is the capsule, which is composed of carbohydrate molecules, making it highly negative and resistant to phagocytosis by neutrophils and macrophages. The tip of the phagocytes is also negatively charged so that the tip would repel the capsulated bacteria. At the sides of the bacteria are appendages like pili, flagella, fimbriae used for locomotion. These appendages are antigenic and can mount a monoclonal antibody response. The bacteria is covered by a thick cell envelope composed of the peptidoglycan and cytoplasmic or cell membrane. Inside the cytoplasmic matrix is the genetic material or nucleoid and ribosomes for protein synthesis. Bacteria also possess inclusion bodies. The nucleoid is double-stranded, circular, supercoiled DNA consisting of one chromosome with a polycistronic messenger RNA. Also seen in the bacteria are plasmids, which are carriers of resistance factors to antibiotics or heavy metals. These plasmids are replicons, meaning they undergo replication without the aid of the chromosome. Plasmids may be transferred to another bacterium through conjugation via the sex pilus or through transformation, which is a genetic exchange through uptake of naked, foreign genetic material. There are antibiotics like streptomycin, tetracycline, and chloramphenicol which bind to units of the ribosomes, rendering a halt in the protein synthesis. This effect is bacteriostatic in nature. They just stop protein synthesis and do not destroy or kill bacteria. The cell wall is composed of cross-linking of peptidoglycan monomers, which is thick in the gram-positive bacteria. The monomer is composed of a unit of N-acetylglycosamine and N-acetylmoramic acid linked by a pentapeptide. The cell wall confers shape to the bacteria and protection from the osmotic pressure of the environment. The antibiotic penicillin destroys peptidoglycan by binding with bacterial enzyme transpeptidase, which cross-links monomers to polymers during repair. With the cell wall destroyed, the bacteria is killed by osmotic pressure of the environment. The actual killing effect of penicillin confers a bactericidal effect. Lysozymes in tears of man have antipeptidoglycan activity. On the other hand, autolysins of bacteria are needed to destroy the cell wall for growth and sporulation. Over the years of using penicillin, bacteria have developed beta-lactamase, an enzyme which destroys the beta-lactam ring of penicillin and its derivatives like amoxicillin, conferring resistance to these antibiotics. Here is the simple illustration of a peptidoglycan monomer. Slide 19 gives us the difference in the wall of gram-positive and gram-negative bacteria. Let us look at the gram-positive wall. The peptidoglycan layer is thick with teichoic acids and lipoteichoic acids, both are seen only in the gram-positive wall. The thick peptidoglycan layer is the basis for the gram-positive reaction, as the primary stain crystal violet together with iodine create a tight cross-linking together which is not decolorized by alcohol. The gram-negative wall is complex, consisting of the outer wall which carries the lipopolysaccharide containing the endotoxin lipidae. Attaching the outer wall to the peptidoglycan are lipoproteins which are alcohol-soluble. The lipoproteins are anchored to the thin layer of peptidoglycan. So in doing the gram staining, initial the structure takes the crystal violet, making it blue or gram-positive. There is no tight cross-linking upon addition of iodine, and furthermore with addition of decolorizer the lipoproteins are dissolved, washing away the outer wall, the peptidoglycan, in such a way that the bacterium takes saffron and the counterstain instead, making it red in color or gram-negative. Here is the structure of the lipopolysaccharide. It has three parts: the O antigen used for bacterial classification, the core polysaccharides again used for classification, and the endotoxin, the lipidae. The lipidae, once released from the gram-negative bacteria, is a potent vasodilator causing shock in sepsis syndrome. Likewise, the lipidae induces release of cytokines, leading to development of spikes in temperature of infected patients. Sometimes during the treatment with antibiotics of the penicillin group, L forms are produced. L forms are bacteria without cell wall, the protoplast being the L form of gram-positive bacteria and the spheroplast, the L form of the gram-negative bacteria. The L forms cause chronic inflammation as they are able to revert to the vegetative forms as the antibiotic is withdrawn. Mycoplasma, is a genus with no cell wall. Therefore, in the treatment with infection by Mycoplasma, the penicillin group of antibiotics are not employed. Slide 22 illustrates the innermost wall of the cell envelope, which is essentially the same with gram-positive and gram-negative bacteria. In the absence of mitochondria in the prokaryote, the cell membrane or cytoplasmic membrane serves as the site for ATP synthesis. The flagellum is used for locomotion. Bacteria with flagella are said to be motile. At the tip of the flagellum, bacteria may change the composition of the surface in a process called phase variation. The development of monoclonal antibodies to the surface of the flagellum due to past exposure may trigger phase variation. Slide 25 illustrates the capsule-making bacterium pathogen. The capsule, being composed of carbohydrates, resists phagocytosis, being highly negative in charge. The ability to form capsule may be acquired by bacteria in genetic exchange. Sporulation occurs in the event that nitrogen or calcium is lacking in the environment of the microbe. There are two genera, the aerobic Bacillus and anaerobic Clostridium, which are able to form antispores. These spores may be viable in the environment for many years. Because they are covered by a hard substance, calcium dipicolinate, they resists high temperatures of boiling. At any time the spore may revert back to the vegetative state. Slide 27 gives us effect of malachite green elucidating the endospore. In the laboratory we will have an exercise with endospores and malachite green. Slide 27 gives us effect of malachite green elucidating the endospore. In the laboratory we will have an exercise with endospores and malachite green. Slide 29 shows us the steps in gram staining which we discussed previously. Here in slide 30 we see the different shapes and arrangement of bacteria. I want to correct that the example given Bacillus anthracis is not gram-negative but gram-positive. In the usual ruling, we say that cocci are generally gram-positive while bacilli are gram-negative. Slide 31 shows us the genus Mycoplasma, which has no cell wall. It is described as having fried egg appearance. The penicillin group has no effect on the bacteria Mycoplasma. Slide 33 gives the usual trait. Bacteria are classified before they are grouped into a classification system. Theses are gram stain reaction, acid fastness, shape and arrangement, carbohydrate utilization, serologic activity and genetic relatedness. Once the traits are known, bacteria are classified into two systems, the Keyes and numerical taxonomy. Keyes makes use of bacterial traits. The Burgess Manual Bacterial Taxonomy is an example of Keyes. Today the numerical taxonomy is the most popular worldwide because of the fast identification. This is used worldwide. In this system a given trait if positive through biochemical reactions is given a score of 5, while in its absence, a score of 0. This system is a static system because microbes evolve and acquire traits through genetic exchange. The other two systems makes use of ribosomal RNA gene expressed as 16S ribosomes in the prokaryote and 18S ribosomes in the eukaryote. The ribosomal gene for these ribosomes are the most conserved gene in the evolution. The other form is nucleic acid taxonomy, which depends on DNA homology. A phylogenetic tree based on ribosomal RNA is shown. Bacteria have 16S ribosomal coefficient while eukaryotes have 18S ribosomal coefficient. In between is the domain with Archaea, which has 16S ribosome. These Archaea group are usually non-pathogens and they are able to survive extremes of conditions. It is said the Archaea are ancient relatives of eukaryotes since they share a common introns, which are regulatory genes in their DNA. Introns are lacking in prokaryotes. The two most important factors affecting growth of bacteria in vitro are the media used, which will be taken up in the laboratory, and temperature. Mesophiles are bacteria that grows well in room temperature, and most of the pathogens of man are actually mesophiles. Slide 37 is a typical bacterial growth curve. This curve may be likened to what is happening in man. The curve starts with a lag phase. As you inoculate and implant bacteria onto the medium, you see no growth initially. In man, this is the incubation period where no signs and symptoms are noted. The next phase is the exponential phase where bacteria multiplies rapidly. Corresponding to the stage in man, it is the exponential phase where the physician appreciates the signs and symptoms. With crowding of nutrients in vitro, a stationary phase is noted where there is an equal quantity of viable bacteria and non-viable bacteria. This phase may be equated to man being given antibiotics. The last phase of the curve is the death phase. This seen in man if the treatment is correct or pre-existing antibodies with immune-mediated response has neutralized the offending bacteria. Thank you for your attention. We will have an open forum and question-answer portion 30 minutes before dismissal. Give your feedbacks to jbromeo@ust.edu.ph. See you in the case discussion. Goodbye.